Oxygen Consumption Rate Fluorometric Assay Kit Operation Guide. Before the experiment, check all kit contents before use. Contact us if needed. Sample preparation. Set up blank wells, control wells, and sample wells in a black clear bottom cell culture plate. Cell seeding in cell culture room. Dilute the cells to the concentration recommended in the manual and seed 100 microliter of the cell suspension into each well. Check cell density and distribution under microscope. Culture overnight in a 5% carbon dioxide incubator at 37 degrees Celsius. After overnight incubation, remove the cells and observe their condition under a microscope. Normal cell morphology with uniform adherence, no detachment or medium contamination observed. Reagent preparation. Dilute the probe 5 to 15-fold with culture medium to prepare working solution. The plate wells are pre-warmed to 37 degrees Celsius before use. Operating steps. Subsequent experimental steps may be performed outside the cell culture room as appropriate. Carefully aspirate the medium. Avoid cell detachment. Add 100 microliter of culture medium into blank wells. Add 100 microliter of working solution into control wells and sample wells. Place the culture plate in the microblit reader or incubator at 37 degrees Celsius for 30 minutes. Add 10 microliter of drugs solvent into blank wells and control wells. Add 10 microliter of drug into sample wells. Immediately add 2 drops of sealing solution to each well. Data processing. Measure the kinetics using the fluorescence microblit reader at 37 degrees Celsius bottom reading. Perform measurements every 2 minutes, with continuous monitoring for over 90 minutes. Set the excitation wavelength to 405 nanometers, and the emission wavelength to 675 nanometers. Data export. Average value calculation. Scatter plot fitting.